Look at any hospital or disaster response system, and you'll see human blood treated as an industrial resource. We run massive donation drives, process the yields on an assembly line, and stockpile the results to keep trauma centers operating. Because getting a transfusion wrong is lethal, we reduce our internal biology to a strict filing system. A simple letter and a positive or negative sign dictate exactly who can save your life. But if perfectly matching this fluid is a matter of life and death, it raises a biological mystery. Why did human evolution leave us with this fractured, incompatible system instead of a single, universally accepted blood type? Zooming in on the microscopic level reveals that red blood cells are often studded with distinct geometric markers called antigens. These proteins act as biological shields, determining which foreign substances the body accepts and which it attacks. But nature rarely hands out free armor. In the microscopic world, every structural shield you build to defend against one threat becomes the exact handle another pathogen uses to break in. Long before agriculture, ancient hunter-gatherers carried what we now call type O blood. It is the baseline model, characterized by a complete lack of A and B antigens on its surface. Moving through ancient mosquito-heavy environments, this smooth surface provided a mechanical defense. The parasite responsible for the deadliest form of malaria needs surface markers to grip and clump red blood cells together. Without A or B antigens, the parasite struggles to grab hold, lowering the risk of severe infection. Yet, stripping away those primary shields leaves the base layer of the cell, a structure known as the H antigen, completely exposed. Certain bacteria and viruses look specifically for that unprotected entry point. If you have type O blood, waterborne pathogens like cholera and norovirus can latch directly onto that exposed H antigen, leading to far more severe illnesses. So the world's most common blood type is a prehistoric compromise. Our ancestors gave up defenses against severe waterborne illnesses to survive the bloodborne parasites of the ancient wild. As humans stopped hunting and settled into permanent farming communities, our environment shifted. We were suddenly living in dense spaces with entirely new sanitation challenges. In response to the bacteria thriving in these settlements, the type A antigen emerged. This new marker triggered a much stronger immune response against the specific infections tearing through early cities. But building an aggressive immune shield requires thicker blood. The physical presence of the A antigen naturally raises baseline levels of clotting proteins. That rapid clotting saved early farmers from bleeding out from injuries. Today, however, that exact same trait translates into a higher statistical risk for cardiovascular conditions and dangerous vascular blockages. Around the same time, across parts of Asia and the Middle East, the type B antigen developed. This regional adaptation altered how the body processes carbohydrates and fats, providing unique protections against local strains of bacteria. Humanity's expansion fractured our internal biology. We traded long-term cardiovascular efficiency for the immediate ability to survive the filth and plagues of early civilization. Only a few thousand years ago, as migrating populations carrying the A and B antigens finally intermixed, a completely new combination appeared, type AB. Medically, possessing every major antigen makes AB-positive individuals the universal recipient. In a trauma ward, their bodies will safely accept blood from any donor without triggering a lethal immune response. Yet carrying every shield means carrying every liability. AB individuals inherit the heightened clotting risks and vascular vulnerabilities of both parent bloodlines simultaneously. Beyond the A and B shields is another marker, the rhesus or Rh factor. Lacking this marker creates a severe biological penalty. If an Rh-negative mother carries an Rh-positive child, her immune system can treat the baby's blood as an invading pathogen, leading to devastating complications. Before modern preventative treatments, this reproductive barrier severely limited how far negative blood types could spread. It is why configurations like B-negative and AB-negative are still carried by roughly 1% of the global population. These incredibly rare combinations represent the extreme edge of human variation. They possess unmatched medical flexibility in hospitals, paid for by deep historical biological fragility. Within the high-pressure environment of a modern trauma bay, blood is valued for its immediate clinical utility. Every bag hanging from an IV pole is a tool used to stabilize a life in the seconds that matter most. But the blood types most heavily demanded by our emergency systems are the same ones carrying our heaviest evolutionary burdens. The broad compatibility of O negative or AB positive are strictly the byproducts of past genetic trade-offs. Your blood type is a highly specific geographic record of exactly what your ancestors had to sacrifice to stay alive. Every drop flowing through your veins contains a microscopic history of the food early farmers ate, the dense cities they built, and the plagues endured. Human survival never required perfection. We conquered the earth through a series of fragile compromises carried quietly inside us right now.